Hello and Assalamualaikum warahmatullahi wabarakatuh. I'm Arif Faisal MUSTAFFA. Today I will present the journal based on genome sequencing project for the subject Stbc3022. The title of the article is Whole Genome Sequencing and Phylogenetic Analysis of Human Metapneumovirus Strain from Kenya and Zambia. The introduction for the human metapneumovirus. Human metapneumovirus is a single-strand RNA virus. They are family from the Viridae related to the human respiratory syncytial virus. They can cause the acute respiratory illness for the young children, and for the size of the genome is around 30 kb. The approach that can provide the valuable resource is first whole genome sequencing approach that can characterize the viral evolution, and they can doing the disease epidemiology, which is to study and analyze the pattern of the disease condition in population, and also to identify the transmission event and outbreak. For the PCR approach, they can target to the whole genome sequencing. Random amplification can amplify the target virus of overall host and environment accuracy. For the methodology, there are two general methodology. First is the sampling of the HMPV detection and genotype assignment. They're using the nasopharyngeal and the oropharyngeal swab sample was collected. They test the sample with the reverse real-time PCR assay to detect the HMPV virus, and the FNG, which is fusion and glycoprotein gene from the HMPV sequence, have been used to analyze the phylogenetic tree, and then they are generate the 5 subgroup from the 5 HMPV subgroup, which is A2A, A2B, and B1 and B2. So for the whole genome amplification and sequencing, there are 5 steps, which is first they are design a primer from the non-redundant yield of the potential primer, and then the higher frequency will be selected, and then they are using the primer mixer. From the primer mixer, they are using the two reaction to avoid the generating the small product of the whole and reverse primer, and then they are going to the viral RNA extraction, cDNA synthesis, and PCR primer, which is the viral extraction. They are doing the reverse transcription process to generate the cDNA, and then the cDNA they are using the PCR mixer to incubate for the several temperature and time, and then the expected product is approximately around 1500 mescaline. For the library preparation, they are prepared by using the Nextra XT kit and a pair with the N sequence with the mixed sequence reagent. This library preparation are used simultaneously with the input DNA in the single tube reaction followed by PCR reaction. And then they will be purified in the library and quantitative and quality check by using the qubit and bioanalyzer. For the phylogenetic analysis, the data of HMPV genome sequencing was being used by the phylogenetic of viral. The data set will be analyzed by using the IQ3 software to infer the maximum likelihood tree that complete the genome and individual gene among the site of the rate heterogeneity. This is the summary of the whole process. Okay, for the result, the genome characteristic and the size of the genome around 30 kb, and overall the identity sequence was analyzed is around 58.2%. The intra-subgroup are divided into 4, which is A2A, A2B, B1 and B2 that share the genome. Also, the intragenic and the nucleotide sequence identity for the HMPV-A and HMPV-B are slightly different. And also the length of the nucleotide consensus around 9 to 90 length, putative start and end of the region flanking to open frame of the viral gene. For the sequence diversity and phylogenetic relationship, all the sequence gene conserve a nucleotide and amino acid level. They are sharing more than 85% between the subgroup and nucleotide identity, and the 90% for the protein identity. There are two sampling that have been collected. For the A2 subgroup from the Kenya, they are closely related to the virus from the USA and the Thailand. And for the B1 subgroup genome from the Kenya, they exhibited 97.5% nucleotide similarity with the virus collected in the Australia and USA. For the sequence diversity and reverse real-time PCR target region, the variant-lifted virus strain may lower the sensitivity of detection, result in the decrease in the quantitative of viral load. New HMPV genome were checked from the nucleotide difference in the genome region targeted by our diagnostic reverse real-time PCR primer and probe used the HMPV detection. A primer and probe template mismatch was identified and one mismatch in the forward region in HMPV group A and one mismatch from the forward and probe target from the group B and 5 different mismatch with the engine based engine based on reverse real-time PCR assay. For this question, where genome sequencing of 5 HMPV strains were determined and compared with the genome published from around the world. Comparative sequencing analysis indicated a fairly conserved positioning of the virus start and end region as well as translation start and codon. The observed sequence conserved in the gene is not usually and is suggestive of the functional and structural constraint on diversity, but less expected of the F gene because the status as a neutralization and protective antigen similarity to its closely relative RSV. And also for the F gene, MIL substantial contribute to cross neutralization and cross protection between the HMPV subgroup. For the G gene, they could attribute to the selective pressure for the amino acid changes coming from the host immunity and ability of the protein to tolerate substitution, which may be due to its purpose extent harmful nature. Phylogenetic incongruence between the whole genome tree and FNG gene tree is reported previously from the HMPV and could be attributed to differential the rate of evolution, selected the pressure, or past recombinant event. As a conclusion, this is the first genome characterized of the HMPV genome from the African patient. Several times of limitation sequence method emission cover for the 5' and 3' trailer region will not capture this primer. However, this method and data provide a useful reference for the design of local diagnostic and study aimed at understanding HMPV epidemiology and their evolution. That's all for me. Thank you.